So we're going to start. So I'm introducing our next speakers, Michal Linial and Dina Schneidman, and they're going to talk about whether the virus has an Achilles heel. So thank you.
Sure.
And sorry, I should just say one thing beforehand, and that's if you're not speaking, please mute your microphone. This is a large meeting, so please do mute. Thank you.
Sure. Okay, everybody here is okay? Good. So good afternoon. It's really a great opportunity, and thanks for the invitation. This colloquium will be a little bit different because we are kind of sharing with Dina and trying to update you with the work and research that we are doing in the last, let's say, 2 months. And because I was asked to talk a little bit to the broader audience, I'll start by a little bit of overview on the virus and the pandemic of COVID-19. So as the title says, does the virus have an Achilles heel? And I would like to change the question and say, hopefully yes, and if so, where is it? And that will be the topic of this discussion. So what you see here is basically the last 2 months of from January 22nd till March, and each line here is a month. And everybody remember that we were all looking at China, and then, in the end of February, we were thinking, okay, China is really affected, but this is what happened a month later. And let me just show you what is the situation at that time, which is the end of March. We had all the world, basically 200 countries with 500,000 people that are confirmed and over 21,000 deaths. 24 days later, the situation is really, really changed. And what you see here is what I showed you in the previous slide. And this is the bump of China, as you can see, really small bump. And what we have actually is a huge increase in these 24 days. And the numbers, as everybody is familiar with, of today or yesterday, we are already have the entire world, 210 countries with over 200 and almost 2.5 million people that are known to be affected, and 160 something thousand deaths. And the good part is that we have already a quarter of the people that are affected or infected that are recovered. And we have this enormous number of over 20 deaths per million globally, the entire world. So this, everybody is familiar with, but I want to show you that there is another epidemic that no one talks about, and this is the infectious trend of publication. The same graph that I showed you before is keep peaking in terms of publication. Most of them are really not worth mentioning, but this is quite shocking to see the trend of publication around us. Now, COVID-19 really provides us more questions than answers. And by now, we know many things, but I'll just mention a few that I find extremely surprising and fascinating. First is the clinical pathology is really complex and unexpected. And physicians are really argue whether they do have one disease or maybe 2 or even 3 diseases that are all called COVID-19. The second point is the asymptomatic people that, or very, very mild symptom. The number range from nothing to 80%. And we don't know yet what the situation is. Age is really the strongest risk factor. You do not need to have a machalot reka. You need to be old in order to be under risk. And there is this gender dependency, which is worldwide, quite amazing. Male and female, the ratio is really 1.5 to 1 in terms of survival. And just recently, a few surprising but really interesting observation in which smoking and tuberculosis exposure might be even protective. So we have to remember the first take-home message: we know this virus for only 3 months. So a lot of things we just don't know yet. So what we decided to do together with Dina was to really intimately learn the virus and really know it. And what I'll be talking today is about the viral evolution, alpha, beta, gamma, and delta, the genetics, 30,000 base, the viral entry, and then I won't talk for the next colloquium. I kept something aside, but then we'll move to Dina that will tell us something about the vaccination and blocking of the virus, and most importantly, how can we tame and control the immune system, which I believe is the most challenging part in the entire endeavor. So this is what I'll be talking about. Now, this is really what we are going to do now, from now till the end of my presentation, is really to go and take the virus, SARS-CoV-2, under the microscope. And indeed, what you see is under the microscope, but really I want to get into the details. So the first question, where did it come from, the origin? Now, in order to answer this question, one needs to use what we call a phylogenetic tree, or some kind of a relation between different organisms and different viruses. In this case, you see MERS, that by now we know that it was the epidemic in 2012, and bat coronavirus and another line of bat coronavirus and so on. So definitely the bats are the resource, the reservoir for many of those coronavirus outbreaks and epidemic. Now, the SARS that was introduced to the human in 2002, it took a few years to realize that this interesting cat, which is called civet palm, or civet, this cat-like, is the carrier and the intermediates between the bat and the human. And poor pangolin, it's not clear that this creature, this very strange creature, is indeed the source for the current COVID-19 virus. So let me take you a little bit like 17 years back to understand how we analyze what is the origin of a virus. So this is a very famous story. I'll tell it in few very briefly. Dr. Liu, which was a Chinese physician, left this city in China and arrived to Hong Kong, to a hotel called Metropolitan Hotel, actually room 911. So for conspiracy, that's a good number. But anyway, he was really a super spreader, which means that he infected already people that flew to Hong Kong, to Hanoi, to Canada, and so on. And the name of the viruses, this is the reason I'm showing you this, Tor means Toronto, Sin means Singapore, and so on. So we could trace back everything that happened from this Mr. Liu, and actually 50% of all SARS cases in the world are connected to Dr. Liu. And so in a sense, what I'm telling you, that it's a kind of a treasure hunt in which you can trace back, because the RNA is changing, the sequence is changing. We can trace back and see who was before, because it's a sequential event. So the trick in terms of bioinformatics or computational is to do sequence alignment and then move to structural modeling. And what you see here is in red, the SARS of 2002. And what you see in red here is the current SARS-2. And as you can see here, they are very similar, but here they are far less similar. So immediately we can understand what happened and trace back the history of the virus. Now, as we all know, there were already 3 events, and using the same methodology that I just explained, we could do the following: take each of those sequences and align them and do the phylogenetic tree. And immediately you see that the SARS of 2002 and COVID of today are rather similar, rather close by. This number 100 means that this node is at high validity. I mean, we have a high confidence for this node. And immediately you can see that even the MERS, the Middle East respiratory syndrome, is from 2012, is a bit like a relative or remote relative. Everything that is shown in this graph is here in this kind of patch. So this is the MERS and the SARS that I just talked about, but the viruses have other members. And what I want to show you that this extended phylogenetic tree is really interesting because something quite surprising and unexpected. Everybody knows that that the virus that is introduced is new to the current population. However, what less people maybe are aware, that there were two viruses here that already infected lung and caused pneumonia for many years already. And actually there are another two that comes from this branch, the yellow one, that also infect young and adult, like kids and adult. And let's remember the name of this red one, which is NL63, because we are coming back to this. But importantly, just note that this is in a completely different genera. So it's a different part of the corona, let's say, superfamily. Altogether, there were 7 events in which coronaviruses had introduced to human. 3 of them you already know are the epidemics, and the 4 that I just mentioned. So let's zoom in into the SARS, MERS, and COVID and do some kind of a comparison. So now we remember that SARS and already was introduced in 2002, the COVID as of now 17 years later, and the MERS was 10 years after the SARS. So everybody that says, well, we are introduced to the first time those coronavirus, wrong. We have 7 members, only the 7th one, the COVID-19, is a new one. Actually, if you look at this table, you see something that is quite amazing. The confirmed infections all over the world in those other 2 epidemics were below 10,000, or in the case of MERS, not more than 2,400 cases. However, the mortality rate was extreme in the case of MERS, was 10% in the case of SARS, and only between probably 1 to 3 or 1 to 2 in the case of COVID. So something fundamentally is different among those viruses, despite what I showed you about their similarity. So let's try to understand this. And my point is, let's try to learn from the old outbreak, what can they tell us about the new one? So the second or the third point that I want to talk about is how fast the virus is changing. And for that, we have to go to history and get an historical lesson. And the history goes about 120 years, really to the, what you see here is the introduction of the epidemic in 2009 that we call swine influenza. Those are all influenza. And what you see, without going into too much detail, is that there is a very intense mixing. So this guy and this guy, this is a human influenza, and this guy, which is swine, this is avian, like a bird, they were all mixed to produce the one that was pretty dramatic in 2009. So my point is that event in which animals are carrier of viruses and they introduced to human occurred for hundreds and probably millions and goes back to Neanderthal and ancient humans. So my take-home message, again, new viruses are likely to emerge, and they do it by big jump. So I'm not planning on COVID 20, 21, 22, but be aware that there are hundreds of bat viruses that are extremely good resource for that. Now, what else do we know about those viruses? We have this I like this graph because it tells us something that covers everything in nature between us, higher eukaryote, and RNA viruses. And what you see here is the size of the genome and how many and the mutation rate of those viruses. And what we can see is that the virus, as I already showed you, the RNA viruses, which is not true for other viruses, as we discussed in depth in one of those papers. RNA viruses are doing a huge shuffling, as I already illustrated for you. They are extremely diverse, as I showed you. They have a short, very short, relatively short genome and very high mutation rate, really. They change like hell. However, surprise, SARS-CoV-2, which is the causal pathogen for COVID-19, has a proofreading a subunit, which is amazing. This is why it can be the longest among all RNA viruses. Now, the biggest experiment that was done in virology was done by air traffic. What you see here is the word and tracing transmission and viral changes over time, over a different part of the world. Now, it's a huge experiment with millions of spreader. Remember what I told you 17 years ago about Mr. Liu. This is many, many millions of Mr. Liu spread all over. And if you go to the virus, what you see here in the bottom is the virus with its 30,000 bases. And I just focused on one case in which I can show you exactly how I can follow the time and see a change in amino acids between leucine to serine. This is one clade and this is another. Now, remember that the virus is doing the experiment. It's not us. The virus already participated in at least 2 million experiments. So it's a huge one. So what can we do with this huge experiment? The NIH collected very nicely many genomes. So I'm just taking a snapshot what happened on March, which at that time 1,500 genome were sequenced. And this is the clade. As you can see, there are 10 clades. You see the light blue and the orange, let's say. Those are 2 of the 10 clades. And what is needed is indeed to connect this data to clinical information, connect to survival rate, and more importantly, connect to drug and drug design, whether a specific variant is more aggressive than other remain to be seen, and it's an open question as of now. But let's move another 24 days, as I did before. This is what happened 24 days after. What you see is that not even the light blue, but the dark blue became dominated. This one became pretty intense, and the orange as well. And you can see in the word that the composition in, let's say, Washington is very different from that in Ireland, and very different also in Spain and in Portugal, and so on. So this is something to be addressed, a very important question. And as I said, it's kind of personalized infection. Which virus infect me or others? One of the last questions that I want to touch before moving to Dina is, how can we get in? So getting in, of course, to the lung. So as I already discussed with you, this is part of this 4 small clades of the SARS and MERS that we discussed before. This is the MERS, this is the SARS, and this is the COVID virus. And surprisingly enough, when we look at this clade, what we see is that the SARS spike, which is the crown of the virus, binds beautifully to a protein called ACE2, which is a protein very abundant in the lung and very important for blood pressure. However, the MERS, which is very similar, as you can see, it's in the same clade, it binds to a different receptor. It's called CD26 or DPP4, and it's a different one. So when we started a month and a half ago, to ask the question that was a question mark. What will be the COVID-19 virus? To whom will it bind? And this is the answer. I'll show you the answer. The answer is really, if you look at the lung, you can see on the top of the virus, I'll just flip it. So now this is the area of the binding. Here's a small part, which is the receptor binding domain. And here is the binding. And that we can do by modeling it. Dina did a very nice modeling that could show that it's ACE2 and not the other that binds very beautifully to this receptor. Actually, if you look from the top, you see something that I find it really beautiful. You see that it's a three type of, this is from the side, the three colors are intertwined in such a intimate way that it really cannot be broken, basically. And this is what happened. This is the lipid of the virus. This is the virus, the spike, and this is the ACE2 on our lung membrane. Now, just after we started this work and had the idea that ACE2 is indeed the relevant receptor, 2 or 3, I think, cryo-EM came immediately into public, and actually they confirmed that it's ACE2 that is the binder, but they also showed something very interesting that we were not aware of too, that there is a situation in which the spike is closed, and there is a situation, as I showed you, in which it's open and then it's exposed its region to the binding. It also showed something quite dramatic, that this Christmas tree is packed with sugar, packed. So all those candies that are on the tree are really sugar that prevents the immune system to attack the spike. Very clever. Now, the approach we took was to do modeling, docking, and molecular dynamics, namely to understand how it looked like, to understand who is the binder, and to understand better what is the molecular dynamic. And two of the observations that were very interesting were the following. MERS, that it has the similar, very similar 3D, as I already mentioned, decided to take a new receptor. But NL63, remember the one that is completely out of the game, has a different structure, still binds to ACE2. So this was something that we didn't expect. And for that, Esther Brill, a really brilliant postdoc of Shai Arkin, was joining the team, and she did beautiful studies on the dynamics of this interaction. And what you can see, without going into too many details, is that the MERS and the NL63 are poor binders. Whenever you are at the left, you are better binder. Whenever you are in the right, you are poorer binder. But more important, look at the difference between our COVID and the SARS. They are all about the same affinity, same strength. However, the SARS is spread so widely and the COVID is pretty tight. So what does it mean? It means that we could ask the question whether the spike and the ACE2 interface is indeed a good target for drug. And from everything that I told you, I want to suggest 3 Achilles heels for the virus. First of all, could we lock the large conformational movement that I showed you? Could we block the spike activation that I haven't shown you, but there is another interesting step of activation? And the main question, can we use and do a shortcut by use the ready SARS antibodies to neutralize the binding of ACE2? And to make it in a more formal way, how different is the ACE2 spike interaction of COVID from the others? And can we repurpose SARS 2002 antibodies, as you can see in the scheme? And the answer is, unfortunately, no. And what you can see here is the COVID in blue. You can see the SARS in red, and the NL63 in green. And what you can see that they are all very, very different. They bind into different region and different area. What does it mean? That the overlap between the three is so poor that it cannot really mimic what happened in the SARS to the COVID to the NL, despite the fact that they are the same. We can show it in many different other ways. This is maybe the best way to show that COVID-19 has two beautiful patch, but SARS in red has only one patch. And if I'm overlapping both two, you can see that they decided to go into different different points in the atomic level of the AC2, which is in gray. So this is really to say that it won't work. So different strategy used by each viruses, and to show it in a very graphical way, what we have shown, and I haven't shown you the entire detail, that COVID-19 has many more binding. Each of them is somewhat less strong. The SARS has more like dynamic. It's kind of a trampoline up and down. So the antibody that we'll use for this guy would not work for this gland. And the NL63 is completely different. So another take-home message, antibody for SARS will not work. And the second one, the interface is the most evolving part. Remember that I told you about this huge experimental in the air traffic. That means that it's going to be evolved very fast. And so I just want to end, this is the last slide, saying that we are looking for the heel, the Achilles heel, but actually the virus is very clever. It's already found some Achilles heels in the lung infected cell, but this is for a different talk. And I'll move to Dina to tell about immunization strategy to deal with this very challenging virus. And thank you very much. So I'll stop sharing and I'll let Dina go over.
Okay. So the question was, can nanobodies help, or can the llama help us in this search for some treatment? So why we are interested in antibodies? Antibodies are a key component of our immune system. If all goes well, when the virus infects us, our immune system should respond and produce antibodies. This is called active immunity. But when the active immunity is not working, we can consider passive immunity, which means we give an antibody as a treatment. And so all the medicines that you've heard of that have a MAB at the end, those are antibody-based treatments, and they are becoming very popular. There are over 80 approved antibodies for cancer, autoimmune diseases, and many other problems. In fact, this discovery of antibodies as therapies for cancer immunotherapy resulted in 2 Nobel Prizes in 2018, one for 5G display technology that enables to do antibody discovery, and the other one for cancer immune checkpoint modulation. So this is very promising, and the number of antibodies in development is around 700 right now, so we will see more of them. So this is how they look like. So antibodies are Y-shape, and the antibody has 2 chains, heavy and light, usually. And the interesting part of the antibody is where you see this circle, ellipse. So this is the difference between all the antibodies in this part. So all our antibodies are very similar, except for the regions that will bind to specific antigen. Now, camelids or llamas, alpacas and camels, they have another type of antibodies, which are called camelid heavy chain antibodies. They are smaller and they only have heavy chains. And they also have this part that binds to the antigens. And we can make it even smaller by cutting the leg of the Y and having something very tiny that we will call nanobody. So now we zoom in on this part that determines where the antibody will bind to. So we call this part complementarity determining regions, CDRs, and those are loops in the protein. Regular antibodies have six loops, 3 on the light chain and 3 on the heavy chain, and the nanobodies have only 3 loops. We call them H1, H2, and H3. And you can see them colored here in different colors. H3 is usually the longest and the most variable loop. And those regions are responsible for the specificity of the antibody. If we do sequence alignment of antibodies, we can see that they are indeed very similar except for H1, H2, and H3 regions that differ between them and determine the specificity. And why am I telling you this? Because the natural diversity of possible antibodies or nanobodies is huge. So there are lots of options, and our immune system knows how to enumerate them to come up with useful antibodies. So why nanobodies are useful? They can reach comparable binding affinities, although they are much smaller, but they can go into the cavities, and they can also go between the sugars of spike protein, if you think about the virus. Because they're small, they're much easier and cheaper to produce and deliver. They are highly stable and soluble, easy to bioengineer. They have better tissue penetration, and the sequence is highly similar to human antibodies. The first nanobody that was approved as therapeutic was approved, I think, last year. So how can we find the perfect nanobodies? There are more or less 3 options. So one is the fudge display technology, the other one is computational design, and of course we can go to llama and give it the antigen and ask for nanobodies. So the fudge display technology mainly limited in the search space. Remember, I told you there are so many possible sequences. So fudge display is basically doing a very local search due to the limited size of the libraries. And computational design is mostly based on known structures, so it is limited to known loop conformations. While llama is free to explore the whole repertoire of possible sequences. This is our llama team, and those are our collaborators in the University of Pittsburgh. So those guys work with the llama and find those nanobodies. Now, don't think they have llama in the lab. The llama is somewhere in Massachusetts and in the llama farm. But what they do have in the lab is a mass spectrometer. And what they do is that they take blood from the llama and they can identify possible nanobody sequences. So the llama produces the nanobodies, and those guys have an incredible technology to figure out which ones are specific to the antigen that we gave to the llama. So they can identify thousands of antigen-specific nanobodies and divide them into low, medium, and high affinity binders, basically giving us a snapshot into immune response. And the questions we ask here computationally is how can we distinguish between low affinity binders and high affinity binders? How the llama basically works on the nanobodies, how it goes from low affinity binder to high affinity binder. And the second question is, what are the epitopes of the nanobodies that we identify? So epitope is the antigen binding site, and it's important to know where the nanobody binds. So I'll talk briefly about each one of those questions and how we address them computationally. So low affinity versus high affinity. So this is something that Liran in my lab tried to do with machine learning. Basically, he trained a model to distinguish low affinity and high affinity binders. So this is the classical problem in classification. And the model worked very well. But what we were interested in is not classification. We want to understand what actually the network learned. And so to do this, what Liran did is basically similar to backpropagation. He iterated backwards to the first layers from the last layer to see which filters are activated. So there are several problems here. What is optimal filter size? Different training runs can lead to different filters. Same filter can be used for classification of both low and high affinity nanobodies. But we invested a lot of effort into actually interpretability of the network. So I'll show you a few examples of what we learned. So this project started before the COVID disease around a year ago. And we worked on two antigens. One of them is human serum albumin, and the other is GST. And our Llama team has identified thousands of nanobodies for each one of those antigens. And we trained two networks, one for each. And here's what we learned. So this is an example of the filter that the network learned, and we can look at that filter and see, for example, that there is a preference for consecutive positive charge, which is shown here, Kr. We have a consecutive tyrosine and glycine. So tyrosines are bulky residues. They insert into the interface, making a very good contact with the antigen. And glycine gives this bulky residues the freedom to adjust to antigen surface. And we see that consecutive aspartic acids are preferred in low affinity binders. So their weight in this filter is negative. And we've found several filters like that that can give us interesting information about what distinguishes between low and high affinity binders. Here is an example from GST. We can see that this filter with two cysteine residues, it is active in high affinity prediction. So we think that what the nanobody does, it stabilizes the slope with the disulfide bond and makes it more rigid for high affinity binders. So we can learn this from the analysis of our neural network. And now I'll briefly talk about the second part. So what are the epitopes of the identified nanobodies? Now we go back to our spike protein. So what we want is to neutralize binding to the lung cell receptor AC2 shown here in gray. The spike is huge, and the nanobodies that bind somewhere else probably won't prevent infection. So our goal is to target this interface. That's why it's very important to find those nanobodies that will be specific to this interface. So how do we do it? So we do it computationally because there are so many nanobody sequences. We are dealing with thousands of them. So we model the structure of the nanobody. And the problem here is that the loop accuracy is low. So we usually will use several models to accommodate for inaccuracy in loop modeling. And we use protein-protein docking algorithms, which also have problem of low accuracy. But what we will do is that instead of looking at one top scoring solution, we will look at multiple top scoring models, let's say top 10 or top 30 best scoring models. So we dock those two together, and we do it for all thousands of sequences that we get from mass spectrometry. And what we get looks more or less like this. So here each row is the epitope that was identified by docking for a specific nanobody. So each row is a nanobody and the columns are the epitopes. And here we have thousands of nanobodies. If we cluster by the epitope, we can start identifying clusters. So for example, we can see that those nanobodies probably bind to a very specific epitope, and we can find additional clusters in the data. Now, the question is, it's only computational, is it real? So how do we check it? We take a subset of nanobodies, let's say up to 100 nanobodies. They are expressed, and we cross-link them. So those are cross-links. They are added, and they give us information about proximal residues. So mass spectrometry again is used here on the cross-linked sample, and it identifies residues that are cross-linked, and it enables us to do a much more accurate modeling. So if we look at what we get, here you can see HSA with different nanobodies that are mapped with high confidence using crosslinks. We can see incredible epitope diversity. We have around 6 to 8 epitopes here. There is an immunodominance. We can see that many epitopes like to go to this region. Many nanobodies like to go to this region, so those orange nanobodies. We can see a high shape complementarity and also electrostatic complementarity, and also very high specificity. And we can also learn about affinity maturation process. So I'll just finish here, and I'll say that there is one llama in Massachusetts that works on the spike protein right now, receptor binding domain, and we are waiting for the sequences from the llama team. And hopefully we will get some good binders from that analysis. Thanks.
So we have some time for questions, if anyone in the audience has any.
I have a question. You hear me? Hello?
Yes.
Yes. I have a question. I just read yesterday or 2 days ago a message from Montagnier, who is a Nobel Prize, and he said that he found out that the virus has been engineered, and it was probably a mistake that it spread. And I wanted to know this idea whether the virus had been engineered or is natural is a question that it's up since quite a while. But the fact that the Nobel Prize came out with this statement seems more interesting. And what is the difference in the possibility of immunization and therapy if the virus is engineered rather than natural? And while you wait, I have also another question. Could the difference of the reaction and the response of the sickness, for example, in Italy, I'm from Italy, there is, the virus seems to be much more dangerous and killing more people than anywhere else. Could this be a mutation?
Okay. Dina, should I answer?
Yeah, go ahead.
Okay. Of course, it make many, you know, it went to the news, Nobel Prize, whatever. But of course, this is why I put so much emphasis of how do we trace back viruses and their changes. And I should say, I mean, there is no reason to believe, you know, it's very hard to say definitely that something didn't happen. But I'll say it in a different way. Knowing evolution and knowing the diversity of the bat viruses and knowing that we are missing the intermediate animal, we are absolutely, it's very likely that it's a natural event. Now, the reason that it was argued that it's not, it's because something very clever that the virus did, the virus, not the lab. I can maybe show you if, should I show, I can do share screen? Oh, okay. So I can show you just one things that the virus did that makes these people to believe that that's the case here. So what you see here is the virus and the S protein that we discussed about, and it's very obvious that if you are focusing on this area, there is an addition few amino acids that were not in any others. So that was the rationale, saying, wow, this region must be inserted somehow in the lab. However, this region is indeed super important. It allows the virus to be as effective as you can imagine. Now, this happens if you just move left or right, you'll see that in different regions, there are addition and deletion. So there is nothing special here besides of course, what we know, that the virus became very optimized when they do such big experiment. I hope I understand. I replied to your question. And you ask another things about
the fact that in Italy it seems to kill much more than in other countries, whether it be a mutation.
Yeah, I don't think this is the virus. I think there are other aspects that, again, I talk about the clinics, but I prefer not to. But definitely countries like Belgium and France and the UK and so on are affected the same way, despite the fact that indeed in Italy, there is this very, very close contact in a multi-generation type of living and so on that makes a huge difference. But this is more social than clinical.
Question. Can I do a question?
I'll let you go first, Mario.
Who? Okay. I saw that all the approaches to do a vaccine were mainly based on the use of the spike protein. At least all the vaccines that are now running. And my question is, why people don't think to use inactivated virus? So many years ago, when SARS do this experiment, that everybody of us is vaccinated with the SARS protein, it's an inactivated virus, and it's dangerous or more dangerous than this virus. Why there is no any group that trying to do this approach? Not only this, it is known that sometimes this possible antigen, and I worked many years ago with one virus, that when they produced the antigenic protein recombinant was 1% or less active than the inactivated virus. So do you have any idea why people are only working on the spike?
Yeah, so I think only spike maybe it's not a good idea. Spike is changing a lot, and yes, it makes sense to look at the other proteins. I agree with you. Why not to go for inactivated virus? I think the goal is to be smarter and to target more conserved parts of the virus so that vaccine is sort of broad range. If you go to inactivated virus, you sort of have one version only. And it's not necessarily that the immune response will be to the conserved parts of the virus.
I think that the most successful vaccine for inactivated viruses.
Yes.
I think this is the first generation of the vaccines, indeed, and they were successful. We know that. But I'm not sure that nobody's trying inactivated virus.
At least all the list of the
seven
pharma groups, everybody is going to the same idea. Everybody is going to the spike. Yeah. Nobody is trying to do inactivated virus. That in some way I understand. It's very dangerous. But okay, Salk did it 16 years ago.
I think.
So we cannot do it now.
If I can just add one comment, the very successful attenuated live viruses, such as BCG for tuberculosis and so on, was developed for 20 years till it was injected. So I think time is an issue, because to make an inactivated and some kind of attenuated that you can inject safely. Now, this is something that I think many people are missing, and I like to say it loud and clear. Drugs are given to sick people, so you can try to help. But vaccines are given to all people that are healthy. So a mistake in this, or let's say a shortcut in developing an attenuated virus could be really, really dramatic if you use it in millions. So I think time is the main answer as of now. As Dina said, this is first attempt, first most, let's say, fast moving attempt.
I can understand in attenuated.
Offline to give others.
Yeah. Okay.
Michal, quick question. A couple of quick questions for you. I'm Ariel with I24 News. The first one is, you mentioned earlier in the presentation that COVID-19 seems to have a mechanism that reduces the mutation rate, yet simultaneously we see about 10 different strains regionally around the world. One, how does that work? How much do they differ? And is that something that we're going to see differences in the cell bonding, the antigens there? And the second question is probably one for Dina as well. Given that viral replication, there's generally two stages where a virus is vulnerable, one being the bonding state and the other being when the lipid shell cracks open inside the cell's structure, allowing it to replicate. Why couldn't antibody not hit it then?
Okay, maybe I'll start with the first one and you'll take the second. So, I mean, this is a fascinating topic. I mean, because as fortunately, every day there are tens of new viruses that are published, I mean, put in the public database, and people can look at them. Apparently, the mutation rate is really, really surprisingly low, despite the view that I showed you, that it's all around. For example, the spike, the interface, has nothing there. So this area is basically cleaned from mutation. I would say, you know, some virologists that I heard, they say there is no point for the virus to get better. It's already optimized. So there is no pressure. So, and again, the fact that this virus has, I mentioned it in one sentence, has a proofreading, makes it completely control its drift, I would say. So as of now, we do not see the drift. We see something that I showed you, that a very, very different kind of partition of the viruses in different parts of the globe. And I'm really, really using this opportunity to force the, the kupot and the hospitals and so on to share this information so we can connect it directly to the clinical aspect. As of now, we haven't been able to do it.
Thank you, and I'm definitely going to want to follow up. I'm going to send you an email afterwards so we're not wasting everybody's time. And the second question, Dina, is it vulnerable during its replication stage once it's inside the cell to traditional antibody methods?
Yeah, so all the antibodies, most of them, they have problems getting into the cell. So most of them work on the extracellular proteins, the ones that are on the surface. It's not impossible, but it's much more difficult.
Okay, thank you very much.
We have time, I think, for one more question.
Question, if possible.
Oh.
Okay. Is the COVID virus the only one that has a proofreading activity? Second of all, no proofreading activity is ever perfect. And in fact, most other cells have all kinds of elaborate mechanisms to get around mistakes in replication or DNA damage, etc. And so what's its error rate? And third of all, has anyone ever tried to correlate sensitivity to the virus, not only with age, but genetic backgrounds or gene expression or things like that of the host?
I can answer you about the issue of the proofreading. What you see here, I haven't shown you, this is the coronavirus that we are talking about. It is an extremely long, what you see here is all single-stranded RNA plus viruses. And you can see who is number one, coronavirus. Why is it so long? It's only because it has this proofreading. Otherwise, you can see that all the rest can do everything with a third of its size. So indeed, it was an open question for a couple of years, why coronaviruses are so long? And I think part of the answer is because they can manage being long while correcting themselves. Now, as you correctly said, no proofreading is perfect. And actually, there is a back and forth quite a lot. And because of this huge audience of people in the world, we see more and more, I would say, as of now, random walk rather than drift or very selected sub-clone. But we are in such this infection chain that I don't know where we are heading, but definitely it's extremely important topic. So all those 30,000 base viruses are those that have a proofreading.
So let's thank our speakers. I guess it's very difficult to clap in this format, but I'm sure everyone is clapping from their side. So thank you very much. This will be the end of this meeting. Thank you.
Thank you.
Bye.